Hi, my name is Jacob Lacey, and I decided to um, choose my topic of solid state interesting materials on semiconductors, and more specifically, in how they're being used to make synthetic DNA and RNA. So with that, so why is synthetic DNA and RNA? And basically, by the way, they do call these more the scientific literature oligonucleotides. I will be using that word a lot. So why is it important? There are a lot of this has been applied into the medical field. They've been used in this for potential cancer treatment and oncology. For example, there's a study done in 2021 by the Department of Biochemistry at University of Otago, New Zealand. Um, Basically, in the paper, they talk about the results of this. They're showing really promising results for this. However, it hasn't been approved for treatment for people, and cancer is still a very challenging disease. But even more promising, and there has been a drug approved for this that is oligonucleotide based, um, is potential neurodegenerative disease treatment. And the article I want to talk about is a research done in 2020 by pharmacist Frank Bennett and neurologist Adrian Craner. They wrote a paper on the positive effects. And what they do call this specific one is called antisense oligonucleotides. And it's on these neurodegenerative diseases. What that basically refers to is, neurological diseases such as ALS, Parkinson's, Huntington's disease. But the approved drug has been for spinal muscular atrophy, and they think it's just going to get better and more promising results for more of these diseases. And so another application is a better understanding of microbial life. And this is how I actually first found out that oligonucleotides first existed, or how I got interested in myself. So the research I do with Dr. Dodsworth in biology deals with these things a little bit. So I'm going to show you just a quick little two pictures. They're the same picture, by the way. It's just they look different, and it's just because different fluorescent lights. But on the right is a DNA stain of all the living culture of from bacteria in these hot springs in Nevada. And on the left in the red is also the same picture of the same culture, but the left is a oligonucleotide based of the on the species of the specific bacteria, and it's also has a fluorescent molecule attached to it, which causes it to glow and the other ones not to. But anyways, since this was a solid state physics class, I thought maybe it'd be worthwhile to do a little review of DNA, and when explaining about the semiconductors, how it could be, I guess, more simple to explain. So DNA was visually discovered by chemist Rosalind Franklin in 1953 through X-ray diffraction. So I guess a quick little side story about her. She contributed a lot of the work to discovering the DNA structure, but a lot of her work was stolen by her other peers, Watson and Crick, and sadly, she never got the Nobel Prize, but they did. So, yeah. The way she discovered the DNA was through X-ray diffraction. So, if you remember at the beginning of the semester, the X-ray diffraction through Bragg's law. So, basically, if you had like a source of a wave, in this case, an X-ray, and it hits, let's say, DNA, at some point there, it's going to reflect off of it and cause constructive interference. And when that happens, this can be processed as an image, and that's what's shown here. This wasn't like a thing done right away either. This was like multiple pictures. There's actually a famous picture called Photo 51. Yeah, and that really took helps discover what that structure was. And there was some other work being done at the time too to kind of show what the DNA is made out of. And once we figured out what DNA was made of, and it was pretty simple, as you can see over here, that was just, it's phosphate backbone, and even more simple, it's 4 nucleotides that 2 of them pair with each other. Like adenine will pair with thymine, and cytosine will pair with guanine, but you won't see guanine pair with adenine. And you can kind of see that over there. But basically, that simpleness of it kind of revolutionized and made a lot of biology a lot more like improved. So I guess we're reviewing RNA. I won't go to this because it's pretty much the same thing, and it kind of almost feels like they're copied, but structured first discovered. It was discovered 4 years later by a biophysicist, Alexander Rich, through X-ray diffraction as well. Picture looks a little different, but I guess the difference between RNA and DNA is not much, other than instead of thymine, RNA has a nucleotide uracil, and RNA looks kind of like a single DNA, but it functions actually a lot different. DNA is more of a genetic material, and RNA is more for making things like proteins. So how does this relate to oligonucleotides? Or how does it relate to semiconductors? So I'm going to talk about semiconductors and how they work, so I can kind of explain how they relate to oligonucleotides. So how semiconductors work. So basically, you have 2 different types of materials that have an imbalance of charge, mainly through they have something called a p-n junction, where the p type will have something called a valence band, which will essentially have holes, or think of it as lack of electrons. And n type will have the pretty much the electrons. Because there's a lack of electrons and electrons, the electrons will kind of want to flow in the direction where there's not, or the holes, to kind of, I guess, think of it as fill. But since this is like a Fermi level fill, it won't take too long before this is essentially filled up, and that kind of shown this illustration there. And once that's kind of filled up, nothing really interesting happens after. However, though, if you introduce a voltage source, or I guess a way you can introduce electrons into this semiconductor, this will kind of remove the equilibrium and will make electrons flow continuously in one direction. And the way the electrons are flowing naturally, I guess, would be the forward bias, which is more common. And then you can make it reverse the other way, called the reverse bias. So how are these things made by nucleotides? Well, the study I read, done by Glenn McGall and his team, showed they basically had a bunch of semiconductors next to each other, which can basically be regulated through the charge. All these semiconductors had an individual photoresistor next to them. And a little bit of review of a photoresistor, it's an electrical resistor that changes its resistance through the amount of light intensity, usually decreasing the more intense the light is. So that's a lot of words. I guess to pretty much think about it in the terms of earlier, those 5, mainly 4 for, I guess if you talk about DNA, not RNA, those 4, you can pretty much set the light to 4 different settings. And that way the way it works is, originally I wanted to do my project on DNA, but I guess, DNA itself is more of an organic thing and not a solid state thing. So the way it's worked, it's through a chemical synthesis, but those set charges will allow to, I guess, change the base. So let's say you wanted to make like an adenine or a thymine or TCG, and you just be changing the light. And then it wasn't really explained well, or how often you need to change the light. It was more about the procedure of it, but the results in this was pretty interesting. So these are two graphs. One was kind of they call it a resist bilayer process, but you can think of it more as like the expected. That's what they call it in there. The expected. So this is like some kind of DNA containing molecule, and it's what it means by expected. And B is the synthetically made one with the photoresistors and semiconductors. As comparison of that molecule, they're they should be looking exactly the same, which isn't, which is pretty much right. Like, there's a few places where the intensity is more or less, but relatively it's the same way. And the error in here wasn't really mentioned too much in the paper, other than the semiconductors were pretty well to adjust to air. But I guess the technology for making these, so these oligonucleotides do peak about 1 micrometer, or you can think about it as 1 million sequences of those nucleotides I was talking about. For comparison, human DNA is about 1.8 meters long. DNA is actually condensed a lot, but if you took one DNA strand and expanded it in like one dimension, it would be about like 1.8 meters long. So we're like a long way to go for this, but this is a lot of the applications are specific gene based, where it's not as long and can focus on small, short strands like that. There are a lot of promising things, and I don't see too much like of the cons. I did see like someone like, oh, what if someone wanted to make a pathogen that was made from oligonucleotides, and that made you just think about it, but it would be just as easy to, I guess, create a cure for the pathogen right away if you could also make a pathogen. So I guess you could fight fire with fire, but I guess that pretty much sums up my presentation. Here are the references I use. So throughout the presentation, I put my sources in brackets. So if you want to go back or look at the video or see the sources I used, feel free to like look at these and read more. But yeah, that's pretty much it. Thank you.